So we have been talking about cadherin molecules. Cadherin molecules of adjacent cells help two cells stick with each other. So that's the function of basically extracellular domain of cadherin molecules. We have also said that the cytoplasmic domain of these molecules plays a very important role, and that is they basically attach the different cytoskeletal elements to that domain, and cytoskeletal elements of the row of these cells, once it is linked together via these adhesion molecules, can provide great resistance to tensile forces. So we are going to now see how these cadherin molecules are attached to the cytoskeletal elements. So let's look at that. Catenins are the molecules that link cadherin to actin. We saw some proteins in the desmosomes, which proteins were hooking up the cytoplasmic domains of cadherin molecules to intermediate filaments. Now let's look at what proteins link cadherin molecules to actin filaments inside the cytoplasm. Catenins are highly conserved cytoplasmic tails of classical cadherins, the type which are, for example, found in adherent junctions, interact indirectly with actin filaments via intracellular anchor proteins which are called catenins. This interaction is essential for efficient cell-cell adhesion. Cadherins that lack the cytoplasmic domain cannot hold cells together in a stronger way. So although cadherins, if they're in a functional form, which means also that there's enough calcium there, they will still stick with each other in homophilic fashion, but they will not be able to have robust linkages between two different cells. Non-classical cadherins in desmosomes intermediate filament and not with the actin filaments. Now we are going to talk about how the actin filaments are linked with these cadherin molecules. Cadherin molecules are indirectly coupled to actin filaments by the anchor proteins alpha-catenin and beta-catenin. I hope you remember again that we have talked about beta-catenin when we were talking about adenomatous polyposis coli signaling in our previous course. So here we have our cadherin dimer. This cadherin dimer, as we have already seen, that has three different domains. The domain for this purpose, for our current purpose, is the cytoplasmic domain, which we will be most interested in. Here, two of these cadherin molecules are coming together within the same cell. This is the extracellular part, this is the intracellular part, and this is the transmembrane part of the cadherin molecule. So these cadherin molecules associate with three different proteins, important proteins. One is P120, which basically binds to cadherin's cytoplasmic domain, and it is thought that it regulates the abundancy and the activity of cadherin molecule. So it will allow the cadherin molecules to bind its counterparts more effectively if P120 is there attached to the cytoplasmic domain of this cadherin molecule in an active form. Also, two molecules of catenins, two molecules each of alpha and beta catenin, they also interact. Here you can see alpha catenin is directly interacting with the cytoplasmic domain of cadherin, whereas the beta catenin is interacting with the alpha catenin of which is directly attached to the cytoplasmic domain. Beta catenin then basically directly interacts with the actin filaments shown here and links this molecule with the actin filament. The cadherin molecule is indirectly linked with actin filament, and we have said that this linkage is important in order to form robust cell adhesion junctions. So the second important role, again, as I said, of beta-catenin, we have already talked about in signaling, when we were talking about signaling. Adhesive activity of cadherins is regulated by cell. At any given moment, a cell might want to stick with its neighbor, but it may be useful for the body, or cell may have a function that requires it to move from one place to another. So the only way cells can do that if they have evolved mechanisms to activate or deactivate their sticky molecules. The sticky molecule in this case we are talking about is cadherin. So this regulation is important for cellular rearrangements which happen very frequently while during animal development, for example, because cells ultimately have to form different shapes, different structures. Initially, all cells are linked together. These cells have to de-link with each other. We have seen how that happens and form other structures. This process of activation or inactivating a sticky protein, cadherin, for example, may involve phosphorylation or dephosphorylation of one of those anchor proteins that we talked about, for example, P120, alpha-catenin, and beta-catenin. Cadherins not only help cells stick with each other, they also transmit signals into the cellular interior. For example, vascular endothelial cadherin or VE cadherin not only mediates adhesion between endothelial cells, but is also required for endothelial survival. I have mentioned this before, I think in one of my previous presentations, that one of the ways our body keeps check on cells, but that cells do not detach from one organ and go to a different organ and start growing there, is by ensuring that extracellular matrix, we'll talk about extracellular matrix later, but basically those cells can only survive, a cell of a particular organ can only survive in the organ it is supposed to be. It cannot detach. For example, liver cell cannot detach from liver and start growing in the space of a brain cell. Although, for example, endothelial cells that do not express vascular endothelial cells that do not express VE cadherin, they still adhere to one another via N cadherin, but they cannot survive for the reasons I've just mentioned. Endothelial cell survival depends upon extracellular signaling protein called vascular endothelial growth factor or VEGF, also referred to as VEGF, which binds to the receptor tyrosine kinase. It uses VE cadherin as a co-receptor. So these molecules, cadherin molecules, are not only again helping cells stick with each other, but they're also providing cells with important signals that they are allowed to live in a particular place. So we have seen how intermediate filaments are linked to the cadherin molecules. Now we have seen how actin filaments are linked to the cadherin molecules in, for example, adherent junctions.